Okay, next speaker is Jelle De Vos from Research Institute for Chromatography here in Belgium, and also a major champion, I would say, the organization of really pushing 2D liquid chromatography forward in industry. So, floor is yours.
Thank you, Dwight, for your kind introduction. I'd like to thank the organizers to give me the opportunity to present some of the work we are doing at RIC. And talk of today is, of course, focused on multidimensional liquid chromatography and on how we push the limits of chromatography for biopharmaceutical analysis. And being indeed at the multidimensional chromatography conference, we all know how hyphenation and coupling columns is the way to do that. So this is a nice picture that always shows how beautiful nature is and how also in biopharma, we humans, our ingenuity comes to play with all these processes inside the cell to cure diseases, to basically have a good impact on our health. And there are several ways of doing this. Of course, monoclonal antibodies is one of the most popular therapeutics these days. But we should also not forget the nucleic acids and the cell-based therapies, which are more and more emerging these days. Today, I will be focusing on monoclonal antibody biopharmaceuticals. Of course, Keytruda is still, at least last year, was still one of the most popular, or actually the most popular biopharma product, being immunotherapeutic antibody. So monoclonal antibodies is still always in the top 10 list of sales of pharmaceuticals. And that is because these monoclonal antibodies really have a huge therapeutic potential in solving a lot of diseases. But unfortunately, from a, let's say, production and storage perspective, these molecules also come with a great structural complexity, often arriving from the biosynthetic process, but also, as I mentioned before, from storage. That's particularly true when we compare to the classical small molecule drugs, where monoclonal antibodies really are large molecules, 200 to 500 times larger than classical small therapeutic compounds. And I said, unfortunately, that's sometimes an issue. Fortunately for us as analytical scientists, that's a challenge, and we like to do that. Typically, how we tackle these monoclonal antibodies, it's looking at different angles. We use one-dimensional liquid chromatography and mass spectrometry to look at the peptide level, the protein level, the glycan level, at subunit level, and there try to really understand these molecules much better. But in recent years, there has been indeed a trend to move to multidimensional analysis to look at these biopharma products. Like here, what has been done is coupling 2 dimensions to boost the resolving power for these separations. Or what can also be done is to couple 2 dimensions to improve the hyphenation with mass spectrometry, as has been shown here with multiple hard-cutting methods. And what I would like to show today basically is how we at our lab have been using these multidimensional chromatography methods for several biopharma applications. And the first one is a 3DLC MS multi-attribute analyzer, where from the cell culture supernatants, we analyze on multiple dimensions. In the first dimension, we have a protein A affinity chromatography column, where we can say something about the antibody titer. In the second dimension, we have a multi-method option to choose between size exclusion chromatography, cation exchange chromatography, and hydrophobic interaction chromatography. And after that, we go to the mass spectrometer. And like that, in this platform, we can simultaneously say something about different size variants, charge variants, hydrophobic variants, and even about the molecular weight, the amino acid sequence, and post-translational modifications, and all of that starting from the cell culture supernatants. This is how it looks in the laboratory, or at least on the picture, how it looks in the laboratory, and a bit more schematically also shown here on the slide. Indeed, multidimensional chromatography, liquid chromatography, it's combining a lot of different pumps, different detectors, different valves. Specifically here we have combined the active solvent modulation valve, a column selector valve in the second dimension to really be able to select between these different second dimension analysis, and then a multiple hard cutting valve to move towards the third dimension desalting column, to send fractions to the mass spectrometer. One example of this 3D LC-MS approach is shown here for a tocilizumab clone selection program. So tocilizumab is known as Actemra or Roactemra, which is basically an IgG1, a humanized IgG1 actually, which works on the IL-6 receptor and is used to cure rheumatoid arthritis. More recently, it was also shown that for COVID, this can be helpful to reduce the so-called cytokine storm. So huge potential for this monoclonal antibody. And in this clone selection campaign, you really see here the power of this multi-attribute analyzer and multi-dimensional analyzer. You see on top our titer information from the different clones. At the bottom left, you see cation exchange chromatography for all the different clones compared to in blue the originator. And on the right, you see nicely the deconvoluted mass spectra for these different clones. And immediately, I circled it already, you see for clone C, there is on cation exchange, there is a shift towards the basic region in the chromatogram, which also in mass spectrometry we could immediately see this linked to 68 Dalton mass shift. So what is the origin of the 68 Dalton mass shift? For that we used 2DLC, high and low pH 2D DLC, to look at the different peptides, and we could see that this 68 Dalton mass shift is effectively a substitution that happened right there in the heavy chain from phenylalanine to isoleucine or leucine, which explains this 68 Dalton mass shift. Like that, what could then also be traced back to the manufacturer, that actually a point mutation was at the source of causing this substitution. Of course, a point mutation there in that part of the antibody is something you typically do not want, because this is really at the CDR region. And when we looked at ELISA of how functional this antibody still was, we could really see a 4 times drop in potency for this clone. So a very good reason to exclude this clone from the selection campaign. Now, we evolved this multidimensional chromatography setup to also incorporate online let's say an online digestion dimension on this multidimensional analyzer. And for that, basically the trick that we did is right here, where we incorporated first a second dimension reverse phase cartridge to desalt, denature, and reduce the protein before sending it to a trypsin digestion column, which you see in blue at the bottom. And after that, because we have here a third dimension isocratic pump installed, we can basically use after digestion a solvent switch, go to more acetonitrile to elute columns of the trypsin cartridge and send them towards RPLC analysis and towards the MS detector. And how we can show the potential of this multidimensional platform is shown here for trastuzumab, which is commercially known as Herceptin, which is used to cure breast cancer. And you see here nicely for the main peak from cation exchange, you see that we get on this setup over 90% sequence coverage. And the nice thing about this setup is that not only nice sequence coverage is nice, but it also allows us to probe in the setup more label modifications, which typically if you would do an offline in solution digest overnight, you could easily miss or introduce, some modifications with typically on this online setup because digestion times are in the order of 20 minutes on our trypsin cartridge. You can have, let's say, much more sensitive detection and of certain modifications. Here's an example, what you see for the pre-peak on the cation exchange chromatogram. We see a deamidation occurring. In the post-peak, we see iso-D isomerization occurring, which is something that has been described already for trastuzumab in literature to occur. But here we see this all on this multidimensional platform, all in one go, all from one sample. When we were developing these multidimensional platforms, we were typically talking about 3DLC and now moving in these platforms to 4DLC type of platforms. Why 4DLC? Because each dimension where something is happening inside our setup, we pictured as a different dimension. But in fact, more correctly, this would be a multidimensional analyzer because not all the different dimensions are really analysis dimensions. But still, for pictorial reasons, it's nice to have a good illustration or depiction between the different dimensions. And what you see here is another modification of this multidimensional analyzer, where we combined actually both setups from previous slides. In the first dimension, we added a multi-column selector valve to be able to choose between different analysis or chromatography methods. And at the back end of the multi-analyzer, we added a split, which allows us to send part of our protein before digestion to do middle-up analysis on the mass spectrometer. And then the last part is still the same as previous setup, where we can still do online digestion. This is how it looks in reality, how the data is showing in reality. You can really see the different protein level and peptide level data coming in from this multi-dimensional analyzer. An example, in this case, this is for a therapeutic FC fragment, which is developed to cure autoimmune diseases. What you see here in black is the reference sample, and in red is an aged sample, which from mass spectrometry, from the middle-up analysis, we could see that the more basic peaks in our cation exchange first dimension analysis picked up oxidation. And actually what is strange is that this normally oxidation does not change the charge of our molecule, so we should not see a shift occurring on cation exchange, but in fact we do see that. So we investigate a bit further and did hydrogen deuterium exchange of those two samples, And you see from the uptake plot on the right that really there is also a structural change occurring because of this oxidation in the antibody fragment. And actually, it's a structural change that exposes different peptides at the surface of our antibody and thus causes a change in retention time on cation exchange. Now, all of this was about focusing on the structural analysis of our antibody, but actually we also want to know, is our antibody still functioning? And for that, the recent trend is using affinity chromatography, particularly for this type of antibody because it's targeting the FcRn receptor. FcRn affinity chromatography is a powerful method to see if the antibody is still functioning. What you see here is in black chromatogram of the H sample where really there are a few peaks appearing on the, let's say, the left, the low affinity range in the chromatogram. And when we couple this to mass spectrometry, again, we could see this oxidation, the 16 Dalton mass shift for the H sample, which is at the cause of this affinity shift. So affinity chromatography is a powerful method to really say something about the function of your antibody. So we wanted to develop a multidimensional platform where really we have both structural and functional characterization of these antibodies in one platform. And these last few slides are focusing on that. So what we did at first is we used cation exchange chromatography in the first dimension and couple it to this FcRn affinity chromatography column with a multiple hard cutting valve in between to send fractions to that FcRn affinity column. And to increase our sensitivity on the mass spectrometer, we also have the option to introduce a denaturing solvent at the back end. This is what we see on the chromatography. So when we take the main peak of, in this case, a full antibody, or full-length antibody, I mean, then you nicely see one nice peak on FcRn chromatography, and also here the deconvolute mass spectrum with the nice mammalian glycans pattern appearing. Now, if we introduce also H samples, in this case a forced oxidation sample with hydrogen peroxide, then we really see on the FcRn affinity chromatography more zones appearing. We called them A, which is basically the non-stressed zone, and then B and C, more stressed affinity zone, where C is the most heavily affected zone in affinity chromatography. And from mass spectrometry, again, we could see that the zone C, which is more heavily affected in function, is indeed the double oxidated variant of the antibody that is appearing. The problem is with this type of setup is that you do not know exactly where this oxidation is occurring, on which part of the antibody. And here we further analyzed these stressed samples and at least show a bigger overview of the previous slide, or a more detailed overview, I mean, where you also see that for a stressed sample, for different charge variants that we are sending to FcRn chromatography, you also see subtle changes in retention time on FcRn affinity chromatography. But I said it already in previous slide, the problem is at this stage that we do not know where all these oxidation events are occurring. And for that, we developed a modification of the multidimensional platform. Sorry. sorry. Well, stuff happens in multi-dimensions. I guess also on the computer stuff can go wrong. But final slide actually. We developed this multidimensional platform to really also pinpoint really where in the antibody, in which part our modification, in this case an oxidation, is occurring. And for that, we had to incorporate also this trypsin column, so this bottom-up peptide analysis approach inside the multidimensional analyzer. And without going into too much detail to lose too much time, this really allows us to truly couple all the retention times in FcRn chromatography to the mass spec data to really give us a way of like a scoring chart. Which antibody species do we see on our multi-attribute or multi-dimensional analyzer, sorry. How does it look like in reality, and how is it functioning? And that's an important message here, that really also the functioning information or functional information of the antibody is scored on this multi-dimensional platform. And with that, I would like to thank my colleagues at RIC, especially Lisa, who is doing all the hard work here for this research, colleagues from industry and university, and of course you for your attention.